I'm going to have to give this talk in English. My Spanish is really too poor. I want to say that I'm absolutely delighted to be here, and I want to thank Benito for inviting me. And so I'm just going to introduce myself briefly. I'm a professor at Johns Hopkins University. I'm a molecular microbiologist with an interest in extremophiles, so microorganisms living in extreme environment. And in particular, I also have an interest in astrobiology. So before I start, I just want to let you know about an initiative that we have at Johns Hopkins University in collaboration with the Space Telescope Institute, which is the institute who's in charge of the Hubble telescope. So we have started about a year ago an institute called Planet Life in the Universe. Sorry, I want to show you this first. Yeah. And so here's the website if you're interested. We have a lecture series. And if you go to the website, you can see all the speakers that we've invited so far, speakers coming. And I want to show you this because even though you're here in Chile, you can go to the webcast page and you'll be able to see the talk. They've been recorded, the slides have been recorded, so you can go and see all the talks that have been given in the past, and also while the talks are given, there is a simultaneous webcast. You can see the talks live. And I can give you the website again later if you're interested. So before I start telling you about what we found about the microbial ecosystem in the Atacama Desert, I want to tell you why we're interested in the Atacama Desert. And one thing is that this is one of the driest places on Earth. So just on its own, it is very interesting to try to learn more about the microbial community, the diversity of this community, the distribution in the desert, and what those microorganisms do, how they make a living. Now, the other interest is that the Atacama Desert is one of the places on Earth that is closest to places outside of Earth, and in particular Mars. So as a Mars analog environment, it's also a very important environment that we should study if we want to inform our search for life on other planets. If we are looking for biosignatures on Mars, on places we can go in our solar system, this is where we should start. This is where we should understand the ecosystem to try to find out what biosignatures we might find on similar environments outside of Earth. It's also very important because we need to be able to find biomarkers that we can remotely be able to detect, and in particular spectral analysis of planets for which we don't have the means to go to. And I'm in particular thinking about exoplanets. This is a very big field right now, very hard. We will not be able to go to those planets in our lifetime, but we can get spectra, we can get information for those planets. And to be able to interpret those signals, we also need to get an idea of what places very similar on Earth, what is the functioning of those ecosystems, and what kind of signal we can get from those environments. So this being said, what do we do in the Atacama Desert? So as I mentioned, we're molecular biologists, and so we use molecular methods. And so what we do, we get samples, we extract the DNA, and we get this environmental genomic DNA. And then from this, we try to get information about the microbial community in the ecosystem. And we use two major ways. This is called 16S ribosomal RNA surveying, where we are particularly interested in a piece of DNA, which is the 16S ribosomal RNA, which is a piece of DNA that is found in all living organisms. And from this small piece of DNA, we can get a lot of information about the microorganism present in the system. We can also sequence all the DNA that we found in the environment and get even more information, in particular about how those microorganisms make a living. So far this is most of the studies that we've done in the Atacama, just looking at the 16S ribosomal RNA. The speakers before me already talked about different places where you find life on Earth. So I tried to summarize this in the slides, which I call the Atacama ecosystems, and places where you and Jacek just talked about this earlier. There are also quartz rocks that can be colonized by microorganisms, and UNITO has been part of those studies. Another environment are those rock varnish, which are the very thin layer that can cover rocks in which microorganisms have also been detected. The gypsum crust that Jacek talks about just now, and this is actually from his most recent paper, 2011. And I showed you this because I want to emphasize the fact that in this gypsum crust, what Jacek found was a combination of eukaryotes, so algae and fungi, which are shown here, and then cyanobacteria and heterotrophic bacteria. These are the prokaryotic. So we have in this very small ecosystem a representations of all the domains of life as we know them. And then the sort, which is more of a type of the studies I'm doing at the moment in the Atacama. So I want to start on telling you a little bit about some sequencing data we got from the gypsum that Jacek gave us to study, and then some of the data we have on the soils. So we got two samples from Jacek. We got a gypsum rock from Girocalar, right? And then, correct me if this is not right. And then another rock from Valle de la Luna, which is more of a calcite type of rock. So what we did, we extracted the DNA from those rocks. We looked specifically for these genes I was talking about, the 16S autosomal RNA genes. We sequenced a large number of those, and then we tried to get some identification about what are the type of microorganisms colonizing this environment. Now, I mentioned earlier that in these rocks there are also eukaryotes, but we've been targeting prokaryotes, so bacteria, more particularly. So what you see here is the relative abundance of a whole collection of microorganisms that we have found in those rocks. And what I've done here is compare lunar rock with tidal rock, and you can see that they are significantly different. The different type of minerals found in different places. Are those places far from each other?
Not very far.
Not very far. Right, but you can see that what seems to be important here is the type of minerals that you have in the rock rather than the geographical location in the desert. So what you see here is the legend is a little difficult to read and you have to start from the bottom, but we see that those rocks are mostly colonized by cyanobacteria. Those are phototrophic microorganisms. They are the primary producers, the basis of the ecosystem in those rocks. And then in particular in lunar rock, you have those actinobacteria, some Danicococcus, and pretty much the same group of microorganisms that have been found in other type of ecosystem in the Atacama, the halides, and also the quartz, and the soil as well. So because we were using 16S ribosomal RNA, we were able also to pick up signal from the chloroplast of the algae. And here's a representation of a different group of algae that we found in the two rocks. And you can see again that there is a different type of distributions of those microorganisms. Now, what is very interesting is that those algae and the cyanobacteria that we found on the rock are very close relative cousins of microorganisms that are typically found in aquatic ecosystems. So then what we did, we took some of those sequences from the cyanobacteria down here and then built a phylogenetic tree, which is basically this tree that shows the relationship between the different type of microorganisms. So this is a little bit complicated, but what I want you to focus on is those purple boxes here. Those are the cyanobacteria that were found in the halide that Jacek just talked about. And then I also have here the cyanobacteria boxed in green that were found in the varnish, the rock varnish. I was unable to use the data from the quartz rocks because the fragment of 16S that we sequenced can overlap with ours. And there are also those names here that you can see, which are microorganisms that are known for which we have sequence information. And then if you focused on those blue arrows here, they represent the cyanobacteria that are found in the tidal rock, and you can see that they're closely related to those found in the varnish, whereas the cyanobacteria found in the lunar rock seems to be more closely related to themselves in particular, and then these organisms that Jacek was talking about. So this is very interesting because it gives us an idea of a relationship between those primary producers, those microorganisms, colonizing the different ecosystem that we found in the desert. So now I want to switch to the soil. We came a few years ago and sampled in 3 places, Bear Hill, and 2 places that we named because we didn't have a name, Kevin Garden and Andrew Garden, and those are names of my children, so this is to name the places. This is Alfonso who was here also. We collected a large number of samples on transects in those different places. So I'm going to try to summarize what we found. So the first thing that you've heard many times already is that there are a very low microbial load in the desert. There's an extremely low number of cells per gram of soil. Now, what we also did in addition to all those molecular methods, which are very important because you can really only cultivate about 1% of all the microorganisms in the desert. So if you really want to have a good overview of the diversity of those microorganisms, you do need to use molecular methods. But we also did some isolation, and we isolated about 100 strains of the desert. And this is a tree that placed those sequences, those organisms, so you can see ant here, and then we have some calf here. Place this in the context of the relationship with microorganisms that we know. And the bottom line here is that the isolated microorganisms were mostly spore formers. So you're talking about dormant microorganisms in the soil that were able to revive. So this is our first clue that the microbial community in the soil might not be very active because the organisms that we can reach to are organisms on the dormant state. So we did quite a bit of sequencing. So those are the samples from the Atacama Desert. Here are the lunar rock and the tile rock. And here are samples from Antarctica. We have 2 ice samples and soil samples, and I just put this here in terms of comparison. So this is the number of sequences we obtained for each of those. So I'll remind you, this is a piece of a Cincinnati ribosomal RNA gene. This gene is common in all organisms. But we have about 25,000 sequences. And so what you do when you get so many sequences, it's a little bit overwhelming. So you start to make little piles. So we basically grouped organisms that were sequences that were very similar in different piles, and we call those piles OTU, operating taxonomic units, just a very technical term that represents basically the level of species. So this is the number of species that we can observe in those samples based on the sequence of a small segment ribosomal RNA. And then we can also estimate how much species we have in the soils using some algorithm that can calculate, give us a prediction. And so what I want to point out here is that most of the richness has been found in, I meant to have this box down here, has been found in the lunar rock. So again, here's our second clue. The richness in terms of the number of species that we can find in the desert is not in the soil, but it's in those very small macro-oases, as Jacek pointed out, those little ecosystems that we can find. So we can also look at what we call the microbial diversity. Everybody has been talking about microbial diversity. What does that mean? So you can calculate index of microbial diversity. And those are important because you can then compare between environment, different type of environment. And so the microbial diversity is a measure of the number of different species that you have, but it also measures the number of members that you have in each of those species. And you combine all of this in a very nice equation, and you get those index of microbial diversity. So it gives you an indication of how many microorganisms you have, but also how evenly they are distributed in those different groups. So if you have an ecosystem where you have one dominant microorganism and a few others, you're going to have a certain type, a certain index of biodiversity, which is going to be different than an environment where you have an even distribution of microorganisms in the different groups. So we compared here the Atacama soil with the lunar rock, the Tila rock. This is Antarctica, and this is from the literature, which are soils from Antarctica, and also a soil from Arizona, which is an arid soil in the United States, but it's not really as drastic as what we found in the Atacama. And you can see that the lowest diversity is found in the Atacama. It's very similar to this upper valley in the Antarctica dry valley, which is also very, very dry and similar in many ways to the Atacama Desert. And this is definitely much lower than anything that you can find on the most other extreme environment on Earth. So very low microbial diversity in the Atacama with those peak of diversity, again, in those ecosystems. All right, so this is showing the different type of microorganisms that we found in our endolytic environment, all those samples from the Atacama Desert and then Antarctica. So again, here are the different group of microorganisms. These are the phyla level, and you can see that what is interesting here is that there is not a huge difference if you compare the Atacama Desert in Antarctica. So the conditions, the geochemical conditions, the weather conditions are very similar. The big difference is that you have about 40 degrees in temperature differential between the Atacama Desert and Antarctica. And then here what stands out is those endolytic environments, the lunar rock and the tidal rock. So how do we reconciliate all of this, and what can we say about the microbial diversity in the soil of the Atacama? So I want to show you one last analysis that we did, and it's not great. Can you see the names?
Yeah.
So what this is, it is an analysis for Unifrac, and it compares the microbial diversity between different type of environment. It compares the microbial diversity in terms of the different groups of microorganisms that are present, in terms of the evenness of those microorganisms, how they distributed between the different groups, and it also takes into account the relationship between the microorganisms between the different samples. So it is a very deep and precise analysis, and so this is a principal component analysis that displays the results. And so what we're looking for is trend of association between those different type of environment, grouping, clustering of the environment that we could explain by some of the data that we have from those environments, geographical location, temperature, pH, anything you may have measured in those environments. So what we found here is that the best separation is on this vector here, where you see again, so here is the lunar rock and the tidal rock that cluster together, very different type of environment. The Antarctica samples do cluster together, indicating that they are more closely related to each other than the samples from the Atacama, but you can see that there is a big dispersion here. And so really what this means is that whereas with the rocks, we have a selection, a very unique selection of microorganisms living in this environment, indicating that those microorganisms have been selectively colonizing those environments. Whereas here in the desert, in the Atacama Desert and Antarctica, very likely those microorganisms have been deposited by the wind. And you can see they are different between the Antarctica and the Atacama, but they're not specifically selected because there are huge differences in, for example, the temperature between the Atacama and the Antarctica. And we don't see a clustering. So very likely those microorganisms are dormant in the soil, and they're acting as a reservoir. They're not dead, they're just dormant. They're acting as a reservoir of microorganisms for those islands of life that you found. So it's actually my last slide where I'm trying to make this point, is that the soil is the reservoir. Those organisms have been floating for millions of years in those environments, the Atacama Desert, the Antarctica. And they're sitting there dormant because they're completely limited, in particular about the water, but also the organic matter. And then when they have a chance, when there is an environment that can give them a little advantage, there is more water, and I'm talking about all those environments here, those islands of life, then they can selectively grow in those environments, and we can see a selection of those microorganisms. Because when we look at the distribution of the different type of taxa, we see that there is a difference. And then the way they get around the very low carbon that is found in this environment is that you have primary producers, the cyanobacteria and the algae that are actually the basis of the carbon in those ecosystems. So this is all I have to say today. I want to really thank the people in my lab actually doing the work, my collaborators at the Institute for Genome Science who did the sequencing, and then our considerable like Chris Mathieu and Chasing. And I couldn't find a symbol in universities. You have to give me one.